Welcome to this explainer. I'm absolutely thrilled to have you here today. Okay, let's just jump right in, shall we? I want you to think of the human body as this incredibly complex, bustling metropolis. You've got millions of cells going about their daily business, consuming fuel, building structures, and inevitably generating garbage. For this biological city to survive, it has to constantly manage and eliminate its toxic waste. I mean, if the sanitation department shuts down, the whole city collapses, right? So today we're going to unpack exactly how your body's toxic waste management system works by looking at some pretty fascinating biological chemistry.
And this brings us to a really crucial question. Every single day, you're eating protein. Every single day, your body's actively breaking down its own muscle tissue. Both of those processes release hazardous chemical byproducts straight into your bloodstream. So, how does your body prevent itself from literally poisoning itself from the inside out? How exactly do we neutralize those internal threats?
Well, here's our roadmap for figuring that out today. We'll start with the body's nitrogen balance, then meet the NPN waste family. After that, we'll look at urea, creatinine, uric acid, and gout, and finally, ammonia.
All right, Section 1, the body's nitrogen balance.
So, nitrogen is essentially the fundamental building block of amino acids, which, as you know, are the building blocks of dietary protein. To maintain optimal health, your body requires this incredibly strict equilibrium. You can see the stark contrast here between a positive and a negative nitrogen balance. A positive balance just means your nitrogen intake exceeds your output. You're literally building up the total protein pool in your body, which is perfectly normal and necessary during periods of growth, pregnancy, or tissue repair. But look over at the right side. A negative nitrogen balance, that is a major clinical red flag. It happens when the amount you excrete is far greater than what you're taking in. We see this dangerous state in cases of severe malnutrition, wasting diseases, diabetic comas, and severe burns, where the body can actually lose incredible amounts of protein right through the damaged skin.
Moving on to Section 2, NPNs, the waste family.
This is kind of the dark side of protein metabolism. When your body breaks down dietary proteins and your own cellular nucleic acids, it creates a family of nitrogen-rich metabolic waste products, known as non-protein nitrogenous compounds, or NPNs. There are about 15 compounds that make up this NPN fraction, but take a look at the breakdown here. Urea is the absolute heavy hitter, taking up the massive lion's share at 45% of our total blood NPN. Amino acids and uric acid trail at 20% each, followed by creatinine, and finally a tiny yet incredibly potent sliver of ammonia.
Section 3, urea, the heavy lifter.
Since it makes up nearly half of all our NPN waste, your body has to manage urea super efficiently. It's synthesized entirely in the liver, which essentially acts as a chemical processing plant. It takes carbon dioxide and highly dangerous ammonia and binds them together to form urea. Urea is this safe, stable vehicle that travels through the plasma to shuttle waste straight down to the kidneys, where it's safely filtered out into your urine. So the really critical point is what happens when those kidneys fail. Azotemia is the clinical term for an elevated level of these NPN products in the blood. It basically means your body's filtration plant is breaking down. If this isn't caught, skyrocketing plasma urea levels lead to a condition called uremia, which is eventually fatal if the patient doesn't get dialysis.
So, how do medical detectives actually measure this in the lab? They perform a test for BUN, or blood urea nitrogen, using this really elegant enzymatic sequence. First, they drop an enzyme called urease into the blood sample. This enzyme specifically targets the urea breaking it down into ammonium ions. Then they add a special indicator dye to the mix. The dye reacts with those ammonium ions and triggers a color change. Just by measuring the intensity of that color change, lab workers can perfectly calculate exactly how much urea was hiding in the blood. It's brilliant.
Okay, Section 4, creatinine, the muscle marker.
Now, if urea is tied to protein and your liver, creatinine is intimately tied to your muscles. It's produced as a waste product of creatine phosphate, the high-energy source powering your muscle tissue. What makes creatinine so incredibly useful clinically is that it's released into your circulation at a highly stable rate that is directly proportional to your muscle mass. Because that daily release is so remarkably stable, creatine is the absolute gold standard for measuring kidney function. Specifically, it measures the GFR, or glomerular filtration rate. That's the volume of plasma your kidneys can filter over time. And there's a critical inverse relationship you definitely want to remember here. When the plasma concentration of creatinine goes up in a patient's blood, it means their GFR has dangerously dropped. The waste is backing up because the filter is clogged.
Testing for creatinine is fascinating because labs still heavily rely on a bit of historic chemistry called the Jaffe reaction, which was actually discovered way back in 1886. When you mix creatinine with picric acid in an alkaline solution, it creates a highly distinct red-orange color. Modern labs use a kinetic version of this, measuring the exact rate of that color change to turn an invisible waste product into a very measurable red-orange clinical warning sign.
Section 5, Uric acid and gout.
Unlike urea or creatinine, uric acid represents the final breakdown product of purine metabolism. Purines come from the breakdown of nucleic acids, the DNA and RNA inside your cells, either from your own tissue breaking down or directly from your diet. But uric acid has one highly problematic trait. It is notoriously insoluble in our blood plasma, and this insolubility leads us straight to hyperuricemia. This is the tipping point. When plasma uric acid concentrations climb above 7.0 milligrams per deciliter, the plasma becomes completely saturated. This overload can be caused by your kidneys just not excreting enough, or by massive overproduction, often driven by diets rich in heavy meats and alcohol, or even by chemotherapy treatments that rapidly destroy tumor cells, dumping a massive load of purines straight into the blood.
Take a look at the enzyme highlighted in blue here, xanthine oxidase. It acts as the key catalyst, relentlessly oxidizing hypoxanthine into xanthine, and then xanthine directly into uric acid. This is the exact chemical pipeline turning cellular building blocks into the urate waste our body struggles to dissolve. And actually, this is exactly why drugs like allopurinol are prescribed. They specifically target and inhibit that xanthine oxidase enzyme to just shut this whole pipeline down.
But what happens when the pipeline isn't shut down and that totally saturated plasma has nowhere to go? Well, the microscopic chemistry turns into an agonizing reality: gout. Because the blood literally can't hold it anymore, the supersaturated urate precipitates out as these sharp, jagged crystals, frequently depositing right into the joint cavities. Your white blood cells try to eat these crystals, but the crystals literally tear apart their membranes from the inside out. This releases lactic acid, dropping the local pH, which just causes even more crystals to form in this vicious, agonizing cycle of inflammation.
Finally, Section 6, ammonia, the toxic byproduct.
Ammonia is produced by intestinal bacteria and released by your muscles during intense exercise. Even though it accounts for a tiny fraction of total NPNs, it is incredibly important. Free ammonia is a highly volatile,
deadly neurotoxin. A healthy liver has to act instantly to grab this ammonia and convert it into the safe urea we talked about earlier. When the liver fails to do this, the stakes are terrifying. Take Ray's syndrome, for example, a severe condition usually seen in children following a viral infection. It causes severe fatty infiltration of the liver, totally crippling its ability to clear ammonia. In these cases, survival is essentially 100% only if the neurotoxic ammonia levels can be kept strictly below 5 times the normal upper limit. Any higher, and the neurotoxicity can easily be fatal.
So I want to leave you with this final thought. Considering the sheer, terrifying toxicity of our own metabolic waste, from tissue-tearing uric acid crystals to neurotoxic ammonia, how perfectly balanced must our internal chemistry be to keep us alive every single day? Our livers and kidneys are working in this fragile, completely flawless synchronization just to manage this microscopic city's waste. I really hope this explainer left you with a deep appreciation for the incredible, unseen biochemistry keeping you healthy right now. Thanks so much for joining me, and keep